to Reason with Science, where we explore the profound questions that shape our understanding of biology and nature. In this episode, we talk with an evolutionary biologist, Iñaki Ruiz-Trillo, whose research sheds light on one of the most fundamental transitions in the history of life, the evolution of multicellularity. Iñaki is a group leader at the Institute of Evolutionary Biology in Barcelona, where he investigates how unicellular organisms gave rise to complex multicellular life. Our conversation explores the deep evolutionary forces that shaped this transition, the role of cooperation and cell differentiation, and what studying the ancestors of animals can teach us about life's complexity. We also discuss broader questions about biological organization, individuality, and the very nature of what it means to be a multicellular being. Join us as we uncover the evolutionary steps that led to the diversity of life we see today. So, let's get started. So, hi Inaki, welcome to the podcast.
Yeah, thanks a lot.
Yeah, so I've been waiting to talk to you about your work. So most of the people, when they look at animals, they look at it simply like people or other animals. But you look at animals in a different way, right? Like from a different perspective. So how do you sort of study animals, or what is your perspective, especially being an evolutionary biologist?
Yeah, well, I think I look at animals in a similar way than others. The thing is the question. So in our case, what really fascinates us, we love animals, we love animals. We love their behavior. We love the diversity of animals, the different body plans, the embryology. But what fascinates us even more than all these things is how they originated, because at the end, all animals, all animal phyla, they come from one ancestor that was single cell before it became multicellular animal. And that's our question. And in order to address this question, you can only, and I say only very clear, because you can only address it if you compare animals, extant animals, with their closest unicellular relative. So what we do is we work with the closest unicellular relative, which will be like the cousins. So if you compare animals with their cousins, you can infer how was the ancestor, and more or less that's what we do. So that's what we do in order to understand how the transition happened, how it occurred, this transition.
Yeah, so that's quite exciting, and of course we can talk about what are the major transitions in this evolutionary history. What specifically you're talking about as the ancestor of all the animals?
Yeah, I'm talking about that animals are multicellular, but multicellularity has been acquired several times independently on the tree of life. So you have multicellularity in plants, multicellularity in fungi, multicellularity in slime molds, green, red, brown algae, and in animals. It's actually, that's important, it's probably the only major evolutionary transition that has occurred more than one time. So, for example, other evolutionary transitions are the origin of life. In theory, only occur once. The origin of eukaryotes, in theory, with our current data, it only occurs once. The origin of conscience or humans, theory, it only occurs one. The origin of bilateral animals, just one. But multicellularity the transition from unicellular to multicellular organisms has occurred several times. And animals, they are all extant animals, they have are monophyletic. So that means the whole clade share one single ancestor. That ancestor was multicellular, but before this multicellular was a unicellular ancestor. We are concerned, or we want to unravel how was the unicellular ancestor and how was the first animal. Because if you understand how was the unicellular ancestor and you understand how was the first animal, then you can say, okay, this is more or less what happened, or this is important, or that was not so important. So basically, that's what we want to get. How was the unicellular ancestor? How was the first animals? And how it transitioned from one to another?
Yes, but if we talk if you go in more details, what is
Yeah.
the actual difference between, let's say, being unicellular and multicellular in that perspective?
Well, this definition will come. So now, with the data we have now, the first animal will be, by definition, will be the first organism that had obligate multicellularity, and that has some of the characteristics that define animals. So that they have movement, that they have, they have sex, they have embryonic development, heterotroph, all these kind of things. And by definition, the unicellular ancestor, which maybe was not unicellular all the time, which actually that's what we think. We think that unicellular ancestor was not unicellular always. It was unicellular or multicellular, depending on the conditions. That will be the unicellular, just before the transition.
And how would you, for example, define simply like the multicellularity itself? For example, we can talk about unicellular relatives, there are many examples, right? Choanoflagellates, etc. And they do come together. They do form some sort of, let's say, colonies or something, right? But that's sort of different than the obligatory multicellularity, right?
Yeah, that's how I see it. I think the different point is the animals have obligate multicellularity. They also have more cell types, very clear cell types. So cell differentiation, they have embryonic development, they are heterotroph, and so on and so on. So, but obligate multicellularity for me will be the moment in which I will say, okay, if it's obligate multicellularity, I will call it an animal. If not, no. So imagine we get a new organism that is in between what we know unicellular and we know multicellular. For me, I would say, okay, I will call it an animal if it has all these features, including obligate multicellularity, and I will not name it an animal if it's not, But of course, these things can change as well, because the definitions, I think you mentioned also in another podcast, they are very, it's a linear evolution, so it's not something that you can mark very clear. Usually you mark very clear when you don't have enough information. When you don't have enough information, everything is super clear. This is an animal, this is a plant, whatever. And when you have more information, then everything becomes less clear.
We can also put it in different way. What are the minimum requirements to sort of be a multicellular organism, right?
Yeah, so the minimum requirement, here I think, coming back to what you say, so multicellular can be also organisms that don't have cell differentiation, and it's actually some authors call that simple multicellularity. So you have different cells, they stay together, but there is no cell differentiation or not very clear cell differentiation. And then complex multicellularity, that was defined by Andrew Knoll, which be the ones that they have cell differentiation. Okay. So the minimum requirement will be that you have more than one cell. Actually, that's what the name says, more than one cell. So if you have more than one cell, by definition, that will be a multicellular. Would you call it simple or complex? Then you will have to analyze more things.
And what would be those things?
Cell differentiation, in the case of the definition of Andy Knoll, which more or less I like it. But, if somebody offers another definition and I like it, I will change. I don't mind changing my mind.
Okay. And let's go a little bit more into the unicellular ancestors or relatives of animals. So give us some of the examples, like how or where do you find these? You specifically study these organisms, right?
Yeah.
So how do you find these samples? Where do you find these samples, etc.? Like, how do you study them?
Yeah, so we are studying different organisms that are closely related to animals. These are 4 different lineages or groups with strange names like Choanoflagellates, Filasterians, Ichthyosporeans, Corallotricians, but the name is not important. We can call them A, B, C, and D. The important thing is that they are all completely different one to another. So A is very different morphologically than B, and B is very different morphologically than A and C and D, and C is very different than the other 3. This is relevant because then that means you cannot take any of those lineages as a proxy of the ancestor, to say, okay, I'm going to take this one because probably the ancestor was like this one. You cannot do that because they are completely different, first thing. Second thing, they have evolved independently also for 600 million years of evolution. So they are now here. You are looking at extant organisms, but what you want to analyze is the ancestor. So if you have 4 different lineages, A, B, C, and D, and there are common things in A and B and C and D, this will be very important because that will mean the ancestor had those features. Okay, that's why we always believe that taxon sampling, so basically to analyze all those organisms, was very important to understand the transition to animals, instead of focusing on only one.
Yeah. And where do you find most of these groups?
I forgot about this question.
It's okay. It's okay.
So, well, in our case, most of the organisms we have, they were isolated by other researchers. So we don't work too much on quanoflagellates because there is a very good lab, actually several very good labs that work on quanoflagellates, Nicole King in Berkeley, Thibaut Brunet, Powell, Burkhardt, many different labs. So we focus most on the other organisms. And in Tesponensis, for example, there was a researcher, Canadian researcher, that she isolated several organisms. And they were isolated, several Tesponensis, they were isolated from inside animals. They don't know if it's a symbiont or it's a parasite or it's food, but they were isolated from inside animals. And then other organisms like Phylasterians and Calodricians, some of them are isolated from freshwater, some are marine. So there's a huge diversity also of environments and habitats. And we have like a zoo of different organisms, and each one is completely different.
And how do you study them?
Well, we do several things. So so basically what we want is to understand how they are now, morphologically, to compare with animals. First thing, how they are now at the genomic level to compare with animals, and how they are now at the life cycle to compare with animals. Okay, so we want to compare all these things. So basically what we do is we do morphology, we do cell biology to understand the life cycles, to understand the morphology, to understand all of that, and we do genomics in order to get what are the gene content of these organisms. That's what we do. So we have the cultures in the lab, we extract the genomes, we already did the genomes of many of them, we analyze the genome, we compare the genomes with animals, and then we do cell biology. And then the other thing that we are trying to do, more than trying, because in some of them we already succeed, is to develop established model systems. So basically to develop genetic tools in those organisms. So transfection and if possible CRISPR as well. So that if we have, and we have, because we have identified genes that are in theory crucial for animal multicellularity, and we find those genes in our organisms, the next obvious question is, what are those genes doing on those organisms in order to more or less infer what was the potential ancestral function? And in order to know the ancestral function or to know the function of these genes in those organisms, you need genetic tools. So basically it will be cell biology, morphological description, genomics, and established model systems.
Yeah.
For us and for others. So we share the model system with others so that their other labs can use them and do other things.
Yeah, that's great actually. But then how would you sort of understand if there has been some sort of convergent evolution in some of those organisms?
Yeah, if there is convergent evolution, which I think that convergent evolution is actually, to me, is quite relevant. It's probably more frequent than we tend to think. Then you will sometimes detect it. Sometimes it's not clear. You can see things that looks very similar. Okay, that could be convergent evolution. That could be homology. Who knows? Very difficult. But in the case of genes, it's very clear, because if there is homologous, usually it's very easy to detect, and the phylogenies and all of that. And if it's conversion expansion, you also detect it. So there is conversion, and we find both things. We find things that are homologous, we find things that are expanded in animals and expanded in our organisms, and we find things that are only expanded in animals and not in our organisms, and the other way around, expanded in our organisms and not in animals.
Yeah.
There is a lot of different things.
Yeah, but let's define the convergent and the homologous evolution. Like, what do you mean by, like, let's say if you find a feature in this unicellular, relatives, how would you sort of understand that? Okay, this is the convergent evolution, and like, what do you mean by convergent evolution?
Yeah, I mean, this is for me is one of the more problematic things on this question. Okay, as I said, it's relatively okay to define convergence homology with genes. With features, yeah, I don't know. For example, in some of our organisms, we describe they do these multinucleate sinusoids that resemble a lot what happens in Drosophila embryos. Yeah, they look very similar. They also activate some of the genes. Does that mean they are homologous? I don't know. And why I don't know? Because it could be perfectly convergent evolution as well, even if they use the same gene repertoire, because they use what they can. Biology, they use what they can to do same things. And why not? Let's do a multinucleate. I mean, there are multinucleates in all the tree of life.
Yeah.
So features, structures at this level, different is invertebrates, but at this level, I think it's very difficult for me. It's very difficult. I will be very afraid to say this is homologous. I will always hesitate, and I will say it could be homologous, it could be convergent evolution. Totally.
Okay. Yeah. So the convergent evolution would be that it starts from a different raw material and still ends up with the same feature, right?
Same feature, same structure, same behavior, and it could be completely convergent evolution. And sometimes convergent evolution uses different genes, but sometimes convergent evolution also uses similar genes. Because, imagine you want to do movements inside a multicellular. Yeah. Of course, if you want to do movements and 3D structures, what you use are genes in order to move and polarize and to have polarization. Yeah. And those genes are ancestral. So, of course, you use those genes, You use cytoskeleton genes. It doesn't mean that it's convergence. It doesn't mean that it's homologous.
Yeah. Let's go a little bit more into the details of the organism per se. For example, like the animals or humans, we have about 200 different cell types, right? And all of them, they are clonal. Of course, there are more details there, but we do have the same genome in all the 200 different cell types. But like, how would you sort of imagine, or what's your sort of idea of the ancestor of animals? Like, would it be like some unicellular or some single cells coming together, but still have the same genome? Like, or you expect that the first thing would be the cell differentiation, where still the cells are clonal and they stay together? How do you sort of envision this sort of ancestor?
I don't envision, because we don't know it.
Okay.
I think we have to be fair, and we still have a big gap to understand these things. We don't know about first came. What is the first thing? It's either cell type differentiation, as you say. It's obligate multicellularity first. Yeah. More complex embryonic development. It's a more complex genome, bigger genome. It's very difficult to know. We know for sure that the unicellular ancestor of animals had first a genetic repertoire relatively complex on genes involved in multicellularity functions: cell addition, cell differentiation, cell signaling. This we know. We know as well, because we have the data, that the unicellular ancestor of animals also had relatively complex mechanisms of genomic regulation. Regulation of different transcription factors on different life stages, microRNAs, long non-coding RNAs, epigenome features, phosphorylation, differential phosphorylation, alternative splicing, almost all the features that animals have to differentiate one tissue to another. So they were pre-adapted to do that. But what came first and second, we don't know. And I think actually, I think that with the current taxon that we have, we will never know unless we find something, because the gap is too far. There is animals here and the other, the unicellular are too far, there is a big gap there. So I don't think we can get it. But to answer your question, how do we envision the unicellular ancestor? We envision the unicellular ancestor of animals as being an organism that has a complex life cycle. So an organism that can do many things, an organism that can be unicellular or multicellular, depending on the environment. That can be multicellular by clonal division, or it can be multicellular by aggregation, depending on the conditions. And unicellular that can be flagellate or not flagellate, amoeboid, multinucleate. So probably can do almost everything, because that's what we see in our taxa. If you see the different organisms, A, B, C, and D, you see all these things. And all these things are also present in animals. So probably the ancestor could do all these things. And if you imagine, if you are a unicellular organism, there is nothing better to say, okay, whenever I want, I'm multicellular. Whenever I want, I'm unicellular. This is perfect. It's even perfect in our lives. Whenever I want, I'm alone. Whenever I want, I'm with some people, which is the same, Depending on the environment, I will be one thing or another. And I think the unicellular ancestor had this complex life cycle, which was probably the precursors of having cell type differentiation, because you created the system to regulate this transition from one life cycle to another life cycle. They were probably transitioned very fast, and then they had all this genetic machinery to do, genetic toolkit to do all these kind of things. And then maybe eventually they were forced to be multicellular all the time. So the life cycle that was multicellular, it was maybe forced to be there. And then they became the first animal. That could be one option. And how it became, it could be either by a multinucleate, or by aggregation, or by clonal.
Yeah, and that simply begs the question, right? What are the advantages of being multicellular?
Well, the advantages of being multicellular, which has been described by several authors, first is that you are bigger, and if you are bigger, you avoid predation and you can eat more things. The second is that you can also disperse better, because you can create specific cell types for dispersion. And probably the most important is the division of labor, to have different cells types that do different things. This is probably the biggest advantage of being multicellular.
And there is another routine, basically, which is present in many multicellular organisms. That's the embryology, right? So how do you see it happening in this first of the ancestors of metazoans?
Well, actually, it doesn't happen in all multicellular. I mean, I think embryology will be in only 3 or 4 lineages. So it will be fungi, some fungi, it will be in red algae, plants and animals. Not in other multicellular. But anyway, in the complex ones, it seems to be a feature that is there. I think that at least that's what we are seeing with some of our organisms. We see morphogenetic processes that resemble quite a lot what happens in embryonic development. So probably the capacity to do these movements and these morphogenetic processes were there. But at some point, that had to be this very fixed, so that whenever you do this, you always get into the same kind of organisms. And I think we will take a long time to understand this, because I think that's one of the most crucial things to understand about the origin of animals, or maybe it's not the origin of animals, but the origin of embryology.
But some of the features, at least, like the genetic features and some of the proteins that you also described earlier, right? They were already present in the ancestral species, right?
Yeah, so the ancestor had many genes involved in multicellularity functions and also in development, but not all. So, for example, genes that are involved in polarization, TGF-beta, for example, WING, for now, because this can change with new organisms. For now, they seem to be specific to animals. Also, some Hox genes, for example, there is there are some genes that seems to be still to be specific to animals, and which makes sense. I think at the end you are evolving. There is an evolution, and you are acquisition of new genes along the evolution. So some genes will always be specific to animals, of course, and maybe those genes could be very crucial. Who knows? We will have to see that.
But if we start looking at it as like the basic principles driving the development of that particular organism, right? I mean, of course, we start with a single cell, and which develops into the whole embryo. But then there are certain features, right? Like the fact that the cells sticking together, that's your cell adhesion that you mentioned. So that would be something like integrin family and stuff like that, which will keep the cells together, right?
Yeah.
Then it's the polarity of the cells or growing embryo itself, right? That which will maintain the shape of that particular embryo in certain way. And all these routines, this is what you're saying, that all these routines were already evolved, at least, or not the routines, but the machinery involved in these routines was already present in the ancestral organisms, right?
Yeah, I think the machineries, we can call it the technologies to do addition, cell differentiation, cell signaling, most of them were there. But of course, some of them were further expanded in animals. Some of them were fine-tuned in animals to make it more complex systems,
Yeah.
But the precursors of this, I mean, the machineries, even the movement, cell addition is present in unicellular relatives, and integrins are there, cadenins are there, not in all the organisms, but some of them. You have protein tyrosine kinasas, which are very important for communication. They are receptor and cytoplasmic protein tyrosine kinasas, some of them which are homologous to the ones in animals, are also there. Many, many genes. But as I said before, not all, but it has the capacity to do many things.
Yeah. And but still, I mean, from those early transitions, it's not like we directly get mammals or the most of the animals that we relate to in a way, right? We get for quite simple animals. So let's talk about those kind of animals. Like what are those early animals they looked like?
Yeah. So again, the first animal is very important to know how it was in order to compare with the unicellular ancestor. And we still don't know now that. We are not in our lab specifically working on how the first animal was, but of course we follow the bibliography. Now there is a lot of discussion whether sponges or ctenophores are the sister group to the rest of animals, and which by the way, even if it's sponges, it doesn't mean that the ancestor was sponges. Because they have evolved for 600 years of evolution. And if they were ctenophores, it doesn't mean that the ancestor was like ctenophores, like extant ctenophores. But there is some kind of discussion on that. My vision is that things are quite gradual in evolution until they don't, I mean, there is a lot of gradual evolution, and at some point there is something that changes. But of course, you cannot get from a unicellular organism something like super complex. You have to go in different steps.
Yeah.
And usually you cannot get to B without going into A. There is some laws that you have to fulfill. And I will imagine that the first animal was relatively simple. It just makes something to change all the things that we were saying, obligate, multicellular, whatever, heterotroph, whatever. But with many things that were related to unicellular. And actually, that's interesting because if you check the genomes and even if you check the morphologies, the behaviors of sponges, ctenophores, and our organisms, you will see that they relate more than bilaterians, So I think bilaterians, there is another extra step,
Mm-hmm.
But for ctenophores and sponges or trichroplax, I mean, there is cell differentiation, but not so much. Yeah. Some cells can can re-differentiate into something that was not before, this will be more typical of a unicellular organism. There are different features that I think it explains that there was something of kind of gradual acquisition of more complex features.
Yeah.
Yeah.
I mean, maybe for like some people, it might be like a little bit shocking that sponges are also animals, but
they were not for some time. They were not considered animals for some time. So yeah.
At least now we have considered them as animals.
Exactly.
Yeah. But let's describe a little bit about bilateria.
Yeah. What do you want to discuss?
Like there is the fact that with bilateria, it's the body axis which starts to appear, right?
Yeah.
Which is not present in all the other animals.
Yeah, I mean, bilaterian, I think the important thing of bilaterians is that suddenly you have two body axes, anteroposterior and dorsal ventral. And having these two body axes and having the anterior part is what creates the conditions to have also a greater cerebrum, a greater system, organic or organ there that controls. It is the nervous system that controls everything. And the movement is also more clear and all these kind of things. So, and then, I mean, it's logically that once you get into bilaterian, the body, you can see it on when you analyze animals, the body plans of bilaterians are much more complex, and then they diversify a lot. And actually, I think 34 out of the 30, I mean, I don't remember now the numbers, but most of the animal phyla are bilaterians, which it means that there was a big expansion, explosion of body plans. One bilaterality or bilaterian animals appear,
Yeah. So there is clear sort of an advantage of having the axis rather than.
I think it's a clear advantage for having a more controlled nervous system, and in order to be a better eater. If you have these two body axes and you can organize everything better, and then you have something that is anterior, something that is posterior, and then of course you create the gut with the opening and the anus on the other side. Everything is much more complex, and then you can be a better eater. You can just go directly. You can focus more on this. I think it's true that ctenophores are also very good eaters, but sponges, for example, are different. Are more filter feeding. They are filter feeding, and then they take the bacteria. But once you become bilateria, you can do many more things.
Is there any study of looking at predators for like different lineages in that way? Like if for or different groups, basically, that if for certain group the predators are more compared to the other, is there any study like that? Like
I don't know. I don't know. Maybe, but I don't know. I'm not aware.
Okay. So, but there is another thing that, of course, like increasing size to some extent may also bring other type of competitions, right? In competitions among the group, etc. So how would that affect the evolution in this sense, like especially the evolution of multicellular animals?
Sorry, I didn't get it. You mean competition
between the individuals?
Within the individuals.
Yeah.
Well, I think there is a change of level of. So actually, I don't think it's really a change. It's just that you have a lot of levels of selection. You have the level of the cells, actually, because our cells are also, they try to survive, and that's very clear. And then you have an individual which is trying to survive, and then a population, and then a species. And of course, the more complex you get as a body plan, and the bigger you get, you have some problems that you have to fulfill, but also you can occupy novel niches, you can eat more things, So I think there is always, like an equilibrium of these kind of things, the advantages and the disadvantage of being bigger or being more complex and things like that.
Yeah, because I was sort of comparing, I mean, this is really hypothetical now, but I was sort of comparing it with the transition of eukaryogenesis itself, right? I mean, at least now we kind of we kind of say that either it's archaea or bacteria, but one of them just engulfed the other, right? It's, I think the jury is still out on which one is or who ate who, but like the point being that that there was some sort of, you know, like this kind of transition, right? So I'm just like trying to think if like these different cells, they come together still, like how would they just like, you know, stay together and maybe like form an organism and stuff like that. Are there other examples in the unicellular relatives? Like, do you see um, the, some sort of examples where they also sort of share the food or nutrients in that certain group? Like, did someone try to study this?
No, I mean, I think in the eukaryotes, which I think
Yeah.
most of the people will agree that the archaeas eat the bacteria. That's what happened.
Okay.
I think in that case, symbiosis was probably the most crucial thing there. We don't know whether microbial interactions play a role in the origin of animals. They could have played a role. Probably not symbiosis, but maybe microbial interactions. But we don't know yet. Okay, so for now, this is something that we need to analyze better and see whether it play a role or not. With regard, I think if I understood your question, you mean, okay, what happens if there are several cells that aggregate, for example, you know, and form an aggregate? Yeah. There's always this discussion of, you know, if it's, you can never get complex multicellularity by cell aggregation. Okay, this is in many books, in many papers, that you cannot get complex multicellularity if you evolve by cell aggregation. But how is this observed? This is observed because they check animals, they check plants, they check, you know, red algae, and they say, oh, they divide by clonal division. So, you know, and they are complex. So complex clonal division. And then they look at slime molds, Dithosterium, and they say, oh, they aggregate and they don't get so much complex. But the thing is, you cannot discard that the first animal came by cell aggregation. You cannot discard that. It's true, animals now, they develop by clonal division, of course. And also the unicellular organisms, they can do both things, you know, they divide by clonal division, but they can also aggregate. One of the problems is the cheaters, you know, cheaters because you have the cheaters, you know, and then it's a probable social conflict, whatever. But the thing is, in most of our organisms, they are almost clonal. Even if you have a culture and you have a lot of cells, they are almost clonal. You know, they divide and they are almost clonal. So I don't think it's a big problem of cheaters. And cheaters, you can't survive with cheaters, you know. Everybody survives with cheaters. We survive with cheaters. You know, society survives with cheaters. The problem of cheaters is if it goes too much, you know. Yeah. When it goes too much, then it's a social problem. But a small percentage of cheaters, we survive. You know, from primary school, there were cheaters, one or two, no problem. Secondary school, university, real life, jobs, there is always cheaters and you survive. So I don't see a problem with that.
Yeah. Are there other sort of understandings that we can get from other examples of multicellularity? You mentioned like red algae, etc.
Yeah.
So,
yeah.
Or do you see like similarities, or there are differences between the different like multicellularity phenomena?
Yeah, I think this is something that we need to do. We haven't done yet. So because most of the communities were separated. So the people working on the origin of plants were going to conferences and things for plants. People working on the origin of animals were another ones, and fungi another ones, and so on. But I think now we have arrived to a point in which we have much more data from all of those acquisitions. And I think we should actually come together and analyze what are the common things and what are the differences. Yeah. And I will imagine there will be common things, and I will imagine there will be a lot of differences, because basically you come from different ancestors. So there have to be a lot of differences, but maybe there are common rules.
Yeah.
And we will only know the common rules if we compare all of that. The problem with that is you cannot compare things when you use different frameworks, People that work like us, that work with these unicellular organisms correlated to animals, we have our framework, our way to analyze our organisms. And the people with plants, they have their framework and their way to compare plants and so on and so on. So the only way to do it is, okay, let's come together, let's fix the framework. Let's see, what do we define here? What is the framework? And then we will analyze whether there is common things or not. But we should do it. We should do it. And I'm actually trying to talk with people from other fields of multicellularity to try to make this happen.
That's great. And it's also interesting that I think it's about 16 times, or at least the examples that we have, it's about 16 times where the multicellularity independently evolved.
Yeah, or 23, 24 in eukaryotes.
Okay.
Yeah, there is several, because, and probably there is more. I mean, green algae has already like 10 or 15 different acquisitions of multicellularity. Fungi has several. So there is quite a lot.
And if we go a little bit more into the comparison between multicellular sort of organism and a syncytium, for example, because that's also quite an interesting thing, right? I mean, of course, it's a little bit philosophical as well, because where do you draw the boundary? I think that's what matters, right? I mean, of course, in one case we have clear cellular boundaries, but in the other case, we may not have those clear cellular boundaries, right?
Yeah, I think, syncytiums or choanocytes, which is very similar to these multinucleates. We do have several in our organisms that have big multinucleates, more than 100 nuclei there. I think these are very interesting because they are also widespread in the tree of life.
Yeah.
And they can do also many things because they have a lot of nuclei. I wouldn't call it multicellular because they are multinucleate, but why not imagine that for example, to me, it's not very difficult to imagine that from a multinucleate, eventually you could give rise to a multicellular. And actually, you can even give rise to a multicellular that has a 3D structure, because you can already create a 3D structure of polarization with the nuclei, because each nuclei will have its genome as well, and they have potentially its own expression level of expression levels of different genes. So for me, it's not very difficult to imagine that it can transition. I like a lot syncytium and choanocytes, all these multinucleates.
Yeah. But also like the size barrier. I mean, we know that, okay, from just by like or what is the size limitation, let's say, with the aggregated unicellular eukaryotes, right? Like how long these aggregations can be.
I don't know, but we do have one organism that aggregates. We can signal it to aggregate. And if we put the system to aggregate, it can occupy the whole flask. It can make a super big aggregate.
Okay.
Can it make bigger? I don't know. I always, talk in the lab that we should try to make it bigger, and see if it goes out of the flask, and occupy the whole lab and so on and so on. But of course, there is some problems, because if you have a lot of cells, how does the food get into the more internal cells?
Yeah.
And then actually, when we create bigger aggregates, what we see is that usually they become hollow inside. So maybe because then they can get more food,
Yeah.
So yeah, I don't know what is the limit. We should check, but maybe we will be surprised, and they can be larger than we can imagine. And again, this is the starting point. It's not the starting point. It could be the starting point, And the starting point, you start small. You start small, but then you transition to something more bigger and complex.
Yeah, exactly. I mean, I was talking about the size because most of the syncytium cells, they can be huge, right?
Yeah.
They could be like meters or even longer than that, probably.
Yeah.
So if multicellularity offers something about the size, probably we may be able to understand something more about that, right? Just by comparing the two scenarios.
Yeah, no, I agree. Yeah.
But but of course, I mean the question would be then, maybe it's the cell differentiation which brings probably a little bit more into the picture when it comes to multicellularity, right?
I think so. I think cell differentiation, it's very important, because it provides an extra thing there. You can do two things. If you start with two cell types, you can do two things at the same time in the same place. That's very relevant.
So how do you now I can get a little bit more philosophical then. So how do you look at boundary conditions then, thinking about, I mean, of course, organisms, organs, cells. So do you think about it, or like how do you sort of grasp this idea of the boundaries or in these systems?
Yeah. Yeah, we do think about these ideas, especially the boundary between unicellular and multicellular, as we mentioned, cells, tissues, organization, 3D structures. I think it's not easy. I think we have a tendency to put things in a box and say, okay, this is this, and this is this, and this is this. But in biology, it fluctuates a lot, and it's very linear, all the things. So you have a spectrum of different things, and again, the more you know, the more you feel the spectrum, and the more difficult it is to have boundaries. But I think this is very nice, because this is what biology is. So I think it's very nice to have this lack of boundaries.
But then do you think then maybe we lack in our thinking in that way? Maybe we need like simply a new way of thinking of these systems, the fact, I mean, of course, for example, we can categorize species of organisms based on their genome, right? But now it depends like how much you can really understand just by the genome of that species, right? How much you can understand about their habitat, about their interactions, all those features. So simply about the biology itself, just by looking at the genome, right?
Yeah.
This is just like one example. It could be the same with looking at proteome or other features, but yeah.
Yeah, I mean, I don't know if we need another way, but we need to be open to see, to challenge our own perception. So I think there is an equilibrium between things that we want to detect rules in biology, common rules, and we want to understand the biodiversity. And sometimes these two things together are difficult, because sometimes there is a lot of, I mean, biology, because it's driven by evolution, and it's a historical event. There are many things that are also by chance, it's complete luck. It happened because it happened, and it's very difficult to understand and very difficult to explain, and sometimes there is also an idea that everything that you see in an organism or in a cell is because there is a reason for that. And maybe there is not really a reason. It just happened for something really different, and it's there. But, it's not that the cell was thinking, oh, my God, this should be super cool for me in some years. I will, things happen. And the historical context. And I think we have to be very aware of that.
And that's why I like more. I also like more the synthetic biology approach, because this is where then we start using the tools that we have developed to looking at the biological systems differently, right? And there, I mean, whatever our hypothesis is, we can either prove it or disprove it, right?
So, No, I agree. Also, we have to be careful, because sometimes in synthetic biology, because it's a system, you can also not generalize whatever you find. Maybe it works with that system, and maybe if you go to nature, you will find something different, and then all your theory is totally discarded. So I think you have to. I mean, at the end, this is, I mean, I think it's common sense. You have to combine different things from different projects, and I mean, and different approximations to the question, and then you combine all of these to get some ideas, and then you put a theory, and then you see if the theory stands or doesn't stand.
But what are the tools that you are developing in your lab, considering the static biology or all the different approaches?
Well, we are trying to develop transfection and CRISPR, so in order to see, but this is very important. I mean, the important is the question, okay?
Yes.
Because for me, because sometimes you can have a system and say, okay, I can do many things to this system.
Yeah.
And then I'm going to ask, what can I ask to this system? Okay?
Yeah.
Oh, I can ask this, and I can ask this. In our case, it doesn't work because we don't want a system that we can think what I can ask. I think it's super cool, but it's not our goal. Our goal is we already have the question. We want to understand how unicellular organisms became the first animal. Yeah. This is what we want to understand.
Yeah.
Then, next step, do we have a system? No. We don't have a system. Yeah. Okay, can we create some systems? Yeah, we can create some systems. And that's what we are doing. We are creating several model organisms to understand some things. Yeah. But each model organism is tuned or is good for one specific question also. Not specifically for how the transition occurred, because that's very difficult. Yeah. Maybe you can apply, you can address how, ancestral genes were co-opted. Maybe you can address whether aggregation can work in one way or another. You can address whether morphogenetic processes are one way or another. I mean, you can address these things, but each model for us is for a specific question.
Yeah. So what are the tools?
Yeah, transfection, genetic tools, transfection, CRISPR, in order to knock out genes or overexpress genes and see what they do. This is for some things, and for some other questions, it's better to do experimental evolution. And for some other questions, it's better to do normal cell biology and see what you see there, And for other questions, then genomics and proteomics is super powerful. I think it depends.
Yes. Yeah. And are you also looking for new relatives of the animals?
Yeah, because as I mentioned in the beginning, there are 4 clades, so A, B, C, and D. 4 clades of organisms that are closely related to animals. So one is conoflagellates, more than 100 species. Then we have Phylasterian, only 5 species. Then Ichthyosporeans, 30, 40 species. And then Corallotritium, which is only have 2 known species, okay? So some of them are still poorly known. But we have 4 lineages. We are working with them, not only our lab, but other labs in the world. The problem is, at one point, we saw, as I mentioned before, there is a big gap between animals and their unicellular relatives. We were always wondering, probably there was an extinction going there. There was an extinction. There were missing organisms there that they became extinct and we don't have them. And then some years ago, we decided to see where our organisms are geographically in the environment, more or less what you asked in the beginning, where are your organisms? Okay, we wanted to know because we only got some from the cultures from colleagues. So then we analyzed environmental DNA. Okay, let's analyze environmental DNA, 18S ribosomal gene, which is the gene used for metabarcoding. And once you have an 18S, it can tell you this is an animal, this is a plant, this is a fungi, or it's A, B, C, and D, one of our traits. And then came a surprise. We realized that there is at least 8 lineages more that are closely related to animals and that remain unknown, and that are not A, B, C, and D. So we thought there were 4 lineages. Now there is at least 12. So we are missing 8 lineages to understand the origin of animals. So we believe that we need them in order to have a better interpretation of how was the ancestor. Can you imagine if we knew that there was 8 lineages of Homo something that are missing? I mean, come on, we would say we need to get them, no?
Yeah.
There is 8 lineages. We don't know where, but probably there are fossils here. Well, now we have the DNA. we know where they are, but we don't know how they look like. And we want to look for them. We call them the Magnificent Eight.
Okay.
8 lineages that are close related to animals and that nobody has ever seen.
Yeah. But then how do you find them? This is like Earth is not that small, right?
Yeah. Well, the thing is, we do have the 18s. So, and we know we are now checking exactly where they are. So imagine you have the 18s, you know where they are. So you go there, you sequence again, and you see if it's there. If it's there, then you go there, you design a proof to do fish, and then you do fish, and you see the shape, the morphology, because we don't know how they look like.
Yeah.
And then once you have the morphology, you come back to the same place, you take again, and with the fish morphology, you try to isolate them. It's not easy.
Yeah.
Maybe we will never get one of them, but I think it's worth trying.
It is definitely. Yeah. And just to like elaborate on that, so like how difficult it is. I mean, because it's not like you can take the sample and look at it immediately, right? You'll have to go back to your lab or something.
Well, that's but the good thing is that, I mean, we have to check it more carefully, but we don't need to go to the North Pole or something like that. Most of those lineages are in the Mediterranean Sea or in lakes in Spain.
Okay.
And probably they are in our backyards. The problem is that nobody has seen them. Maybe they are not beautiful, maybe they are not very interesting, and nobody checked them, That can be. And there is also a problem with biology, because we get so specialized.
Yeah.
If you are an expert, for example, let's say you are an expert in quanoflagellates.
Yeah.
If you are an expert in quanoflagellates and you go and you sample things and you bring the samples to your microscope, one of the best things to be an expert in quanoflagellates is to be very good at discarding everything that is not a quanoflagellate. So you don't look at that. You just focus on quanoflagellates. If you are an expert in ichthyosporeans, it's the same. You bring the samples, and one of the best things to make you a real expert in ichthyosporeans is to scan very fast and discard everything that is not an ichthyosporean by morphology. And the same with the sponges, and the same with ctenophores and everything. But of course, we don't have experts on things that we don't know how they look like.
Yeah. That's the conformational bias in science.
Exactly, a little bit, yeah. You go for things that you know how to do it, you like them, and this is your expertise, and then you look for them. And of course, you don't look for things that, and also we didn't know they were there. The thing is, now we know, and if we know, I mean, come on, we have to go for them.
Yeah.
It's 8 lineages.
Yeah. So apart from these 8 lineages, what are the other major questions which are bugging you right now? I mean, not right now, but like in general.
Yeah, no, right now, I know, every day. So another thing that bugs me a lot is whether there are more than 8. Because, and I'm going to tell you why, because all of these we know because we took data from environmental surveys. On these environmental surveys, they use universal primers for 18S. Universal primers. Universal primers amplify all eukaryotes, because that's the idea. You amplify all eukaryotes because you want to know the diversity of eukaryotes. But because they amplify all eukaryotes, there is less chances that you get organisms that are not abundant, which is probably our organisms. So one thing we want to do is sequence with a specific primer for the part of the tree of life that we want. So it doesn't sequence fungi, it doesn't sequence plants, it doesn't sequence bilaterians, and it sequences only non-bilaterians and holozoans. So are there more than 8 magnificent? That's one big question. Is cell differentiation present before the origin of animals? Another big question. And if it is, how it happened, Are the morphogenetic processes that we see on those organisms the precursors for those things that we now see in embryonic development or not? Another important question. Do we have the same rules, as you were saying, they have general rules between different multicellular acquisitions or not? What are the differences? What are the common things? Another important question that bugs us quite a lot is, sorry, I got lost. So, yes, so the different multicellular acquisitions, I forgot. I wanted to say something else and I forgot. Maybe it will come to my mind because I had it here, but now, yeah. Anyway, at least those.
So these are like, and of course, I mean, the next question would be like, what's your sort of approach to these questions? Okay, one thing is that you are already trying to bring different scientists together, so in some conference or something.
Yeah. So actually, we are organizing a conference in October in Barcelona, and that will be about different multicellularity acquisitions. There will be people from plants, from fungi, red algae, brown algae, and animals, of course. So yeah, and then I'm trying to communicate quite a lot with different people from different multicellularity acquisitions. I think it's something that we need to do.
Yeah. No, the other thing that you mentioned about the cell differentiation, I think I saw one paper from Omaya Duddin. I think it's in, I don't know whether it's published now or not. I saw it in bioRxiv where they show the I think they are ichthyosporian probably.
Ichthyosporian.
If I'm correct, yeah. This kind of embryo-like morphology, that was really something, like the whole thing is quite cool, I think.
Yeah, it's quite cool. I mean, actually, I remember still because Omaya was in our lab. So I remember still the first day that Omaya came and said, Iñaki, I saw something that it looks like, at that time we call it, we call it a quartet because it makes these 4 cells, and we were actually looking for, because we saw that in other organisms that was this specific lineage, this specific taxon. But then it stopped to do it, So when we wanted to analyze it, it stopped doing that because sometimes these cell lines, they do these things. Sometimes they do something for years and then they stop doing that. So we couldn't analyze it anymore. And then he came and say, look, this one is doing that. And that's where everything started. So, okay, I think you should check it, because it's very cool.
Yeah.
As I said before, this is morphogenetic processes that resembles. It doesn't mean it's homologous, but it resembles a lot. The important point here for me is whether there is cell differentiation, and it looks like there is already some kind of cell differentiation. And also we have some potential cell differentiation in some kind of facet colonies. There is a paper by Powell Burkhardt that demonstrates that, shows that or suggests that. And we also think there is some kind of cell differentiation in some of our aggregates. So at the beginning, some years ago, you asked me 10 years ago, I would say there is no cell differentiation or not spatial cell differentiation in unicellular organisms or in unicellular relative of animals. Now, I think there is.
Yes. So, and then the cell differentiation, you would define just by the change in the gene expression? How would you really?
Yeah, ideally, morphology or function will be ideal, if you detect transcription factors that are associated to specific functions in the expression, or even a different morphology or different behavior, cell behavior, that will be ideal.
Yeah. And and of course, then there is this another long experiment, the multi experiment from Will Ratcliffe, right? Do you have plans to do something similar with some of your organisms, like the organisms or model organisms that you have in the lab?
Yeah, we've done a little bit in one of the organisms to see whether it evolves into bigger aggregates, and then analyze the bigger aggregates and see what happens.
So this is the one that you mentioned that just covers the whole flask or something.
Exactly, that one. We didn't do this big flask. We just making a selection on bigger aggregates, and then we have the question: what happens when you have bigger aggregates? Do you have more cell types or not, or what happens inside? Some of the tools to make it better, to make a better analysis, we don't have it yet. So maybe we'll have to do more things in the future once we have better tools. But I think this is a very first approximation. I think actually there is an advantage in this organism. There is a disadvantage, very clear disadvantage, if we don't have the same tools as yeast. We cannot do almost anything compared to yeast. But they also have a clear advantage, is that these organisms, they share many genes with with animals. So they have the raw material. So the comparison for me is it's more relevant to the origin of animals, not to multicellularity. To multicellularity, this is already super valid, and you can do many things. But specifically to the origin of animals, this has the advantage, or these organisms have the advantage of having more genes that are relevant for animals, and then you can compare better.
Yeah, but what would be what are the selection pressures you are using?
Well, we are using, this bigger aggregates in this case.
So just the gravity then?
Yeah, exactly. Yeah, okay. Gravity. Okay. I think we could do it in a different way. We have to think more about that and maybe consider other things. We are doing also other experimental evolution projects, but are not so much in the bigger size on the origin of animal, but we also are trying to do whether we are checking now, we are doing experimental evolution, what happens if you grow them in microgravity conditions, what happens if you grow them in low oxygen conditions, what happens if you grow them in without cosmic radiation. There is these special laboratories that they have 800 meters of mountain, so that they have 40 times less cosmic radiation. So we are also doing an experimental evolution there, like half a year growing there without cosmic radiation and see what happens. But it's not totally directed to the origin of animals. It's just different things that we are interested.
Yeah, okay. Makes sense, right? That's at least because you can also get at least some sort of understanding about the principles which we always interested in. So
exactly, yeah,
that's the
and also because if you check, it's not clear yet, but there is many authors that say that oxygen levels where there was an increase at the origin of animals or at the origin of the Cambrian. Some say maybe not. Some say there was a lot of variation. We also know that cosmic radiation was changing also during the evolution of Earth. So we are also checking whether this could be something relevant, different things.
Yeah. Okay. And what are the other sort of understanding that we can get from your work, apart from whatever, like all the things that we have discussed? Apart from that, like what else we can get?
Well, I think the most important message is it's always important to check many different organisms. Taxon sampling is very important. Yeah. One single species can completely change what you were thinking before. So I think that's the main message. And then the other message is we still need to work a lot to understand the origin of animals, and maybe we'll never understand it, but it's worth trying because it will explain a lot about how evolution works and the mechanisms of evolution, the mechanisms of life.
Yeah. Do you think at some point, like, it's again another sort of techie question, since we have a lot of information about AI nowadays and stuff like that. But do you think we can like simulate that part? Do you think we can use like the information that we have and try and simulate at some point? This is now really hypothetical that way, but
I think we can simulate. Okay. Another question is whether the simulation will make any sense or not. Because it's biology again, so it's very historical. There is a lot of things, but I think yes, when we learn more things, when we have better understanding of the mechanistic things that were important for multicellularity and for evolution in general, I think we can model the transition to multicellularity. I think we can model the evolution of organisms now. I think we can model more things. But of course, the models will need to take a lot of parameters, and then this makes things complex. But yes, why not? Why not?
And okay, so if people want to reach you or read more about your work, what's the best way to do that?
I don't know. Check Google Scholar and then see our papers there. We have also a web page that we try to keep all the information there. I'm also in Twitter. Okay. Even now in Blue Sky. So
okay, that's great.
Now in 2 different social media, even if I'm not very fan of social media, now I'm 2 different social media.
Yeah.
So sometimes we are more active, sometimes less active. It depends. But we try to put everything that we do somewhere so that you can find it.
Yeah. I mean, some of the papers that I read, they were super cool. So I really liked it. So yeah, thank you so much for accepting the invitation. This is great. Great conversation. Thank you.
Oh, thanks to you, Jitendra. Thanks a lot. Have a nice day.
You too.